Hello, good day. My name is Ana Maria. My colleagues are Juanita and Maria Manuela. We are biological engineering students of the Universidad Nacional de Colombia. We are currently working in Sustancias Activas y Biotecnología Research Group. And this project is in company with Centro Nacional de Investigaciones de Café, CENICAFé. The title of our poster is Functional and Genomic Analysis of Native Colombian Yeast Strain with Potentially Used as a Starter in the Production of Specialty Coffee. As you might be aware, Colombia is well known for the quality of its coffee. During the fermentation process, the attributes of the final beverage can be greatly improved. Three yeasts have been identified as important in the production of volatile organic compounds. The objective of this poster is analyzing their encoded genes and their involvement in the assimilation of coffee, mucilage, and synthesis of secondary metabolites with the improved qualities during fermentation. Methodology. To sequence the yeast genome, Illumina sequence was used and genome assembly was performed using spades. The phylogenetic tree were 10 constructed and open reading frames identified for each yeast. A blast analysis was conducted followed by genome annotation of the mitochondria in the NCBI. Additionally, a bromatological analysis of the mucilage was performed in order to compare the carbohydrates with yeast metabolomics. The glucosides were annotated, and finally a scanning electron microscope was carried out.
Well, let's talk about the results. In the first graph, identify as Hanseospora varum. In the second one, the Hanseospora genus that show us Unal 103, it's Starmelera genus, and in the top of the graph, it's Starmelera bacillaris, and in the bottom, Starmelera solvosivorans. Here we have the genome similarity comparison. It's important to say that all of the percent identity of each species are above 90. And here are some tests to verify that the genomes of the strain yeast has completely sequences.
As for the mitochondrial annotation, here's a graph showing the mitochondrion of Starmelera sorbosivorans that was submitted with the accession number down below to the NCBI. The second figure shows the growth on the petri dish of the yeasts as well as the yeast during budding in SEM microscopy. The last figure is the starch and sucrose metabolism of the yeasts that was related to the bromatology tests that were done that were trying to find the carbohydrates that are part of the mucilage and the enzymes that were encoded in the genome of the yeasts. Here are the conclusions, especially the metabolism of this yeast is indeed related to the composition of coffee mucilage. The annotation and characterization of the genomes is going to provide valuable insights to better understand the behavior of these strains in the coffee fermentation process, as well as providing a genetic framework for the use of these yeasts. The references are here. We especially wanted to highlight the work of our colleagues Aida Peñuela and Lilibeth Chaverra, whose thesis work are the very basis for this work. of this research. And thank you very much.